Welcome everyone for the first lecture of this particular course of RBC. So the chapter is composition and structure of red blood cells. So we are going to see the complete constituents present in the RBC, its plasma membranes structure and the internal cytoplasmic structure of RBC. So let's start the chapter. So firstly, we will discuss what is the size, volume and thickness of RBCs. So RBCs or erythrocytes are the common name of RBCs because RBCs are red in color. So erythro means red and cytes means cells. So RBCs contain the oxygen carrying protein hemoglobin, which is a pigment that gives whole blood its red color. So very important function of RBCs that they have this hemoglobin with them, and this hemoglobin is something which gives our blood its prominent red color. A healthy adult male is having 5.4 million RBCs per microliter of its blood, and a healthy adult female is having 4.8 million RBCs. Okay, so we can say that 1/4 of the human cells are RBCs, right? But why there is a difference in the number in male and female? Males have higher number of RBCs, whereas females have lesser number of RBCs. Why is it that? Because the production of RBCs is affected by the presence of androgens, which are male hormones, male sex hormones such as testosterone, etc. So these hormones affect the production of RBCs. They increase the production of erythropoietin hormone, and which increases the number of RBCs. Whereas in females, we know that we don't have testosterone hormone, so that's why the number of RBCs is lesser. Red blood cells live for about 120 days in males, whereas 110 days in females. So this is a tentative number of their lifespan. RBCs are highly specialized for their oxygen transport function, the very important function which no other cells perform. This is because RBCs have hemoglobin inside them, right? And for giving hemoglobin a good amount of space, RBCs empty their cytoplasm by having no nucleus, no cellular organelles. And this is why RBC's internal space is increased and hemoglobin gets to take some space over there, right? And uh, and this is to be noted that much amount of hemoglobin and much amount of oxygen carried to the cells. The edges are rounded, thicker than the center. Hence, the center appears to have a lighter shade. Also, we see the dumbbell shape of RBCs when we see it from the side. So, this is the view of RBCs where it is embedded from the center. There is a depression from the center, and it is swollen from outside. Okay, from the periphery. Now, if we see the top view of RBCs and if we cut it from the middle, we see the dumbbell shape over here, all right, and inside the whole RBC, hemoglobin is present, right? So, hemoglobin is filled inside the complete. Mature erythrocyte is a flexible biconcave and anucleated cell, as we have already discussed that RBCs are mature RBCs are anucleated because we want to create some space for the filling of hemoglobin inside it, right? And it is biconcave. So in the previous structure, you can see that due to this indented shape in the middle, it is having this biconcave structure. In all the mammals except camel, RBCs are like this. So camels are having this oval and biconvex and nucleated RBCs, whereas all of other mammals are having anucleated, biconcave and flexible shaped, dumbbell shaped RBCs, right? Due to the early part of fetal life, even the mammalian red cells are all nucleated, but in the later part, nucleated cells disappear from the circulation. That we have already discussed, that nucleus is removed during the maturation of RBC to create some space for the hemoglobin. Red blood cells have a simple structure because it contains only plasma membrane and a cytosol having no cell organelle. All right. Next is plasma membrane of RBCs. So if we talk about the RBC's plasma membrane, it is very simple in structure, but yet very specialized. So we will see what's special in it. So plasma membrane is strong and flexible. It allows them to deform without rupturing as they squeeze through narrow blood capillaries. It is a very important and special feature of RBC's plasma membrane. Why? Because RBCs have to travel throughout the body through our blood. All right, so blood carries RBCs from one part of the body to another through the very narrow capillaries to bigger blood vessels and blood veins and arteries. So capillaries are very narrow, so RBCs have to squeeze through their cavity to move through it. So that's why they should be flexible but strong enough to deform, okay, and they need not to rupture. If they rupture, then all the hemoglobin will be spread throughout the blood, so that's what we don't want. That's why they are flexible yet strong. The permeability of membrane of RBCs is very highly selective. Why? Because no bigger colloidal molecules like any macromolecules, they cannot pass through it without the permission of particular membrane. But some crystalloids like urea and ions, they are freely permeable, so it is freely permeable to these kind of molecules, but the bigger molecules have to take permission or have to move through the gated ion channels so that their movement can be carried out easily. Certain glycolipids in the plasma membrane of RBCs are antigens that account for the various blood groups such as ABO and RH blood groups. So we are well known to to different blood groups like A positive, B positive, AB positive, negative, things like that. So these blood groups, how they get their names? They get their names because of the presence of some special antigens on the RBC's plasma membrane. Okay, so if our RBCs, if someone's body's RBCs are having a particular antigen, suppose A antigen, then its blood group will be A, and vice versa, like for B, for AB, for 0. Okay, 0 for when we having no antigens on the RBC surface. Now let's talk about its cytosol. So RBC's cytosol is very simple because it is lacking a nucleus and other cellular organelles. It doesn't have anything inside it, and it can neither reproduce nor carry on extensive metabolic activities, because for metabolic activities to be carried out, we need mitochondria, right? Mitochondria is the ATP producing cell organelle, but they don't have these, so that's why no heavy metabolic activities can be performed inside the RBCs, right? They cannot reproduce because they don't have nucleus. Okay, so they have a limited lifespan of 120 days. The cytosol of RBCs contain hemoglobin molecules, and these important molecules are synthesized before loss of nucleus during the RBC production and constitute about 33% of the cell's weight. So hemoglobin is something which occupies most of the space of RBC's cytosol. Each RBC contains around 280 million hemoglobin molecules. That's a huge amount, right? Now let's talk about the cytoskeleton of RBCs. So RBC's plasma membrane, if we see, so it's just like a normal normal plasma membrane having this lipid bilayer, but this lipid bilayer is having some special proteins inside, like some integral proteins that we are going to discuss. All right. So the red blood cell membrane contains around, sorry, it contains around 20 major proteins and at least 850 minor ones. So this is a huge number. The integral membrane proteins are organized into macromolecular complexes centered on band 3, the anion exchange channel. So there is a band 3 protein forming an anion exchange channel right through the lipid bilayer. All right, so this is very special integral membrane protein found only in the RBC's membrane. Most of the peripheral membrane proteins form the membrane skeleton, a protein meshwork 40 to 90 nm thick that laminates the inner membrane surface. So here you can see different proteins are also here that are making, that are laminating this inner membrane surface. So in these proteins we have spectrin, actin, and associated proteins like tropomyosin, tropomodulin, adenosine, dimatin, protein 4.1, and ankyrin. So they are like protein 4.1, adenosine, actin, they are forming a complex like structure with the spectrin protein lying underneath them. Okay, this spectrin is very important to this is very important to keep the normal biconcave shape of the RBCs. Okay, so RBCs are flexible, they are easily deformable because of this spectrin protein. So its role is very important. Now, spectrin is bound to the membrane at sites containing the anion exchanger band 3 via cytoskeletal protein ankyrin and adenosine. So ankyrin and adenosine are some proteins which keep this spectrin connected with the band 3 protein, which is an anion exchange channel. All right. Now the composition of RBC. So earlier we have discussed the plasma membrane structure of RBCs. Now we are going to see what's inside the RBC. All right. What is inside its cytosol. So it is having a semi-liquid material inside. Okay, and having 65% water and 35% solids, of which 33% is hemoglobin bound to 2% stromal meshwork of proteins, phospholipids, cholesterol, cholesterol esters, and neutral fats. So these are the components found inside the plasma, inside the RBC. Other organic substances like urea, amino acids, creatinines, adenyl pyrophosphates, diphosphoglycerates, etc. They are also present, but in very small amount. Okay, so you don't need to remember these many things, but yeah, just have an idea about the names of all of that. Of the total lipids, 60% of phospholipids, half of this is cephalin. So cephalin is found in abundance. 30% free cholesterol, 10% fats, and cholesterol esters. Of the salts in the corpuscles, so what is the important salt found inside the RBC? That is potassium phosphate. Whereas if we see outside the RBCs, like in our blood plasma, sodium chloride is highly abundant, whereas inside the RBC, potassium phosphate is highly abundant. Now, there are some variations in the red blood cell count under various physiological conditions. Like if we say diurnal variations, so what is the diurnal variation? Day to day, day and night variations are also there in the number of red blood cells. So this is very interesting. The variations amounting to about 5% occurs in 24 hours. The count is lowest during the sleep. So while you are sleeping, your RBC number is lesser. Then it gradually rises and becomes maximum in the evening. When you do muscular exercises, it raises the RBC count temporarily. Now, while you go to higher altitudes, their count also rises, whereas at lower altitudes, their count falls. Higher external temperatures, so when the temperature is high, the red blood cell count increases. Any condition which lowers the oxygen tension of arterial blood increases the red cell count, and this is normal because when we are having hypoxic conditions, when we are in oxygen stress conditions where oxygen is lesser in amount, then our red blood cell count starts increasing to grab more and more oxygen from the atmosphere, so an injection of adrenaline and excitement increases the count. So adrenaline or any other exciting hormones, when they are injected, the steroids, they also increase the count of RBCs. So this was all for today's lecture. Thank you all.